Well, thank you, Teresa, and it's a pleasure to be back here across the bay and kind of see old professors I worked with and to meet new faculty members. And thank you for all of you for coming to my seminar today, where I'm going to present a little bit of a story that I've been working on in my postdoc towards kind of a new way to make antibodies outside of animals and kind of moving away from having animals do all of our grunt work. And kind of to motivate all of this work, we're really moving towards a new era of biotherapeutics. These can take the form of diagnostic markers, which can tell doctors what types of disease patients have, as well as really new targeted therapies that attack some of the weaknesses of a lot of these diseases, such as cancer. And what's essential to driving these both diagnostics as well as targeted therapies are answering a number of key kind of fundamental questions, namely, what is disease and who has disease? And then when we've diagnosed a patient with a particular disease, what drugs, how should we actually treat that disease? What are kind of some of the Achilles heels we might want to target to develop new therapies? And ultimately, when we're treating patients, we'd like to know maybe a priori whether a particular patient class would respond to a particular drug. Or even more importantly, when we're giving patients drugs, we'd like to know that they're actually responding to the drug in a more kind of real-time fashion as opposed to conventional therapies. And really what's fundamental to all of this is advances in our fundamental understanding of disease mechanisms and what's actually going wrong inside cells in the case of disease. And it's this knowledge that's going to drive kind of the new era of biotherapeutics in the form of improved diagnostics as well as new targeted therapies. And so in order to actually understand disease mechanisms, we must take kind of a step back from even that and understand kind of how cells make decisions and how do cells respond and how maybe this becomes dysregulated in the context of disease. So on a kind of schematic level, cells have a series of cellular receptors that serve as antennae to kind of sense and respond to what's going on in their environment. And these receptors then relay this information through a very complex circuitry to ultimately elicit a cellular response. And this can tell the cells to grow, to die, or to change its state. And really, it's dysregulation of this circuitry that's a hallmark of various diseases, such as cancer, diabetes, and neurodegenerative disorders. And so to really understand what's actually going wrong, we kind of would want a comprehensive picture of what's actually happening in the cell at any given time point. We'd like to measure what interactions are happening, as well as what information is going from the cell receptors to ultimately the decision-making process of the cells. And so not surprisingly, cells have evolved a very complex network of both biophysical and biochemical events to relay information and to make decisions. And these take the form of, can be generally classified as biophysical events, which could involve two proteins interacting to elicit a change in function, protein small molecule interactions. And more generally, layered on top of this is an even more complex series of enzymatic biochemical reactions, which involve chemical modification of these proteins, which often alters their function. And it's these chemical modifications that'll be the focus of my talk today. And namely, you can think of these chemical modifications as the bits the cells are using to kind of both process and store information for the long term. And these chemical modifications are more generally referred to as post-translational modifications. These are different types of chemistry performed on proteins, of which there's over 200 different types. And within kind of our cells at any one moment, it's estimated to be there's over 1 million different types of proteins that are modified by various types of post-translational modification. And these occur at very defined sites in the protein, and they often modulate the activity of the protein. So it could upregulate the activity of a protein or downregulate it. And to further underscore how important this is in the case of disease, our genome encodes for roughly 10% of our genome encodes for proteins that modulate and regulate these post-translational modifications. So these are enzymes that either add the chemical modification or remove the modification. And various cancers are classified by alterations in some of these enzymes. So what do these modifications look like? Well, one of the most commonly studied modifications is known as phosphorylation, the addition of a phosphate moiety to a hydroxyl side chain on proteins. There's also similar small chemical modifications, such as acetylation. And these can gradually get progressively bigger in the case of O-glycinacylation, which is the addition of a single glycan or oligosaccharide, again, to a hydroxyl of a protein, or they could even take the form of large protein domains in the case of ubiquitination. And so one of the grand challenges in the field is developing methods to both detect and quantify a lot of these post-translational modifications, because that's really giving us information about what's happening in particular cells at a given time point. So this could tell us that the protein level is increased in activity or decreased in activity. And so this is the fundamental challenge. And so over the last about 2 decades, antibodies have really begun to revolutionize the study of post-translational modification. And for those not familiar with antibodies, I'll explain in a few slides. But you can think of them as a specialized type of protein that allow us to specifically detect a chemical modification on one protein. And these were first invented about 30 years ago, and they've really enabled our fundamental understanding of post-translational modifications in the form of population-level detection, so being able to identify which changes occur on a global level inside, say, cancer cells, as well as beginning to ascribe function to particular types of chemical modification. And more recently, these have been applied in single-cell level analyses to begin to discover some of the fundamental stochastic nature of various cellular decision-making processes. And probably most importantly to all of this, these have been fundamentally required to enrich proteins which have been modified by these post-translational modifications to then enable our ability to both detect and identify what sites on a given protein are indeed modified. So how have these antibodies been previously made, and what are the problems with it? Well, I'd just like to outline schematically how conventional researchers make their antibodies and kind of to highlight some of the problems with this strategy. Well, the general workflow is as followed. One can start with a target that we'd like to generate an antibody against. So let's say we have a protein which we know is really important, that we'd like a reagent that allows us to monitor that protein. So we can inject or immunize an animal, wait 2 to 3 months, and kind of do whatever it is you do during that time, and then ultimately isolate the cells which are making antibodies from that animal. And then through a very exhaustive screening and production and characterization platform, hopefully identify one single hybridoma or one single cell line which is producing the antibody that we'd like to use. So this antibody will then enable us to detect this protein in a variety of different settings. And this has a number of limitations, as I've kind of touched on. It's very time-consuming. It takes 2 to 3 months just to do this kind of initial step here, regardless of what all this downstream work is going on. It's extremely low throughput. In the best case scenario, you're effectively killing one animal for every new target you'd like an antibody to be raised against. And it requires this kind of black box strategy that is effectively exhaustive screening of lots of different mammalian cells to hopefully identify a cell type that has an antibody that you'd like to use. And finally, more importantly, this is inherently random. So we could basically take one protein that we're interested in having an antibody against, inject 2 animals, and in one case, the animal would give us an antibody, in the other case, we'd get nothing. And so this highlights the fundamental limitation of this method is the process often fails. And even when it does work, so let's say our animal does give us an antibody, we learn absolutely nothing about how to go back and improve this process, other than the fact that, OK, we need to inject some animals the next time through, and hopefully it works that time. So why is this a problem? I've already touched on how for the last 2 decades this has worked. Well, the grand field of systems biology is moving towards kind of a general classification and identification of all of the sites of post-translational modification in human cells. And so using a variety of techniques, researchers have been able to identify for these 6 types of post-translational modification a large number of modification sites. And so I'd just like to highlight this is a logarithmic scale here on the y-axis. And so in total, we have around 250,000 sites of modification that we know are happening in cells. For most of them, we know absolutely nothing about what it's actually doing inside the cell, whether it's altering, whether it's upregulating or downregulating the activity of proteins. And so what's the source of that limitation? Well, effectively, we have essentially no antibodies or tools which allow us to detect and quantify these modifications in a variety of different contexts. And so even in the best characterized type of post-translational modification, namely phosphorylation, we're still almost 2 orders of magnitude under the sites that we know exist in cells. And so there's a huge gap between our ability to functionally characterize and ascribe any meaning to these modifications. And this gets even more daunting when one uses bioinformatic methods to predict all of the potential sites of post-translational modification. We go well into the order of 10 to 100 million sites that could exist. And so really what this highlights is that existing antibody generation methods, which have been around for 2 decades, have really failed to keep up with the biology, and we're missing a good degree of the information that's actually happening inside cells. And this greatly limits our understanding of both cellular responses and then fundamentally disease mechanisms, being able to take a snapshot of a diseased cell and figure out what exactly is going wrong. And several groups have tried to develop like a quantitative metric of this, and so they've performed exhaustive literature searches of publications and patents to, not surprisingly, reveal that most research focuses on a very small subset of proteins. And one common feature of all of these is that they possess well-validated tools and antibodies to enable the detection and quantification of these proteins in various settings. And as one kind of illustration of this, if one basically determines the number of citations for a related family of human proteins and plots on the y-axis the number of citations and on the x-axis just the protein name, you can see clear stratification between proteins where we have an antibody and proteins where we don't have an antibody. And so we effectively have no information about what's happening in the cell to any of these proteins at any given time. And many of these proteins have been shown to be mutated in cancers or other diseases, suggesting they're highly important for medical research. So what's the way forward? Well, I've already touched on our natural way to make antibodies, relying on the natural immune systems of animals. Well, within the last decade, there have been tremendous advances in the laboratory to make what are known as synthetic antibodies. And so these are based on single or a few kind of stable single protein scaffolds that really have excellent biophysical properties. So most of these have melt temps on the order of 80 or 90 degrees. So they're extremely stable. And more importantly, this allows us to make these in bacteria. And so we can grow large bacterial cultures and get grams of these antibodies produced from a liter of culture. And this enables us to rapidly generate these on the scale of 1 to 2 weeks in the laboratory, as well as include and incorporate recent advances in automation and robotics to multiplex and high throughput this process. So now we can generate 96 antibodies kind of en masse using such as colony pickers and liquid handlers and such. And I've done some work in my postdoc developing some of these automated high throughput antibody production pipelines. And finally, most importantly, these are absolutely renewable. So at the end of the day, we have the sequence for this antibody, which can then be digitally encoded and stored in perpetuity for future generations of researchers that can basically pull out the sequence and say, OK, I have an antibody that works in this situation against this target. And you don't have to rely on going back to animals to kind of doing this process again and again and again. So what is an antibody? So the canonical depiction of antibodies is this Y-like structure here. And is a bivalent molecule which has 2 surfaces which can then bind its target protein. And this region is known as the fragment antigen binding region, or FAB, that I'll be referring to. And it's this small fragment of the antibody that we can work with in the lab and manipulate in bacteria. So we can take this FAB protein, which contains the binding site for its target, and produce these in bacteria by putting the 2 genes that encode for the 2 chains of the antibody on a plasmid and introducing this into bacteria. We can then get milligrams to grams level of any particular antibody. And what this also enables us to do is to take advantage of a bacterial virus known as a phage to incorporate the genes for this antibody inside this bacteriophage. And this way, when we make this virus, every virus we get back out has one antibody on the surface and the gene that encodes that antibody inside. So now we can basically do all sorts of manipulations on the antibody to isolate antibodies with distinct properties, and at the end of the day, have the genetic information that encodes for that antibody. And so what does this enable? Well, we can now, using advances in molecular and synthetic biology, make very large pools or libraries of different antibody sequences, incorporate them or package them into this bacterial virus, this phage, to make antibody libraries on the order of 10 billion distinct members. And this is 2 to 3 orders of magnitude more antibody diversity than you or I have in our bodies at any given time. So this has tremendous functional diversity to have antibodies of lots of different properties. So to take this one step further, well, how do we want our antibodies to differ? Well, we'd like to program both spatially where in the antibody, as well as chemically, what types of diversity we include in our antibodies. So we'd like to only selectively change regions in the antibody that contact targets that you'd like to bind, as well as only use amino acids that are more likely to contribute functional chemical diversity. And so we can program effectively where in the antibody as well as what we're encoding. And so we and others have used natural antibody structures and sequences to guide the construction of large antibody pools and libraries with over 10 billion distinct members. So this is all great. We have this antibody pool with lots of different antibodies. What can we do with this? Well, we can take this library and perform what's essentially a binding reaction against a target that we'd like to have an antibody against. And we can wash away all the antibodies that don't bind to the target, so this population, and then selectively elute the antibodies that have bound to our target of interest. And so we obtained an enriched population of antibodies that bound our target. And then since this is in this phage, we can quickly amplify this in under a day in bacteria to develop a new library, which can then be further selected and enriched for high affinity and specificity binders against our target of interest. And so since this is all in vitro, we can play a number of games. Namely, we can reduce concentrations of both the target as well as the antibodies, as well as incubation times to really drive the identification of only antibodies that have very high affinity and specificity for our target. And at the end of the day, after a number of iterations, we can break these phage particles open, sequence the gene encoding for that antibody, and then perform downstream characterization and ultimately application of this antibody. And so our current synthetic antibody libraries are highly successful against stable, well-behaved proteins. So we can routinely obtain single-digit nanomolar antibodies against pretty much most protein targets. However, the main challenge in the field has been, for technical reasons, we have to make short synthetic peptides to develop antibodies against post-translational modifications. And this is more of a technology issue in that it's much easier to chemically synthesize a peptide with a defined phosphorylation event on it than it is to make that same protein with a modification at a single site. So this introduces a number of challenges into the system. Namely, there's a much smaller surface area for the antibody to inherently bind to in this case, as well as the inherent high flexibility that this peptide possesses. And so it has a high conformational entropy that it must give up upon binding its antibody. And ultimately, there are fewer side chains or chemical handles for the antibody to grab onto in the case of this short peptide, suggesting we need highly optimized binding to obtain an antibody against a post-translational modification. And so at the start of my postdoc, I asked the question, well, can we use something, basically knowledge of our target, to guide the rapid generation of synthetic antibodies? So in my case that I'll be talking about today, I knew all of my targets were modified by a single phosphorylation event. And so they all had a phosphate group on them. So how would this work kind of in a grand scale in practice? Well, let's say we had a protein that we were interested in studying with a number of different types of post-translational modifications. I've touched on how right now we and others are using generic stable antibody scaffolds to routinely generate antibodies against the protein itself. However, these inherently completely fail against any of these sites of post-translational modification. So wouldn't it make more sense if, let's say in the case of this green oval, which is a phosphate group that we'd like to target, to engineer and develop an antibody scaffold which has a predefined binding pocket, which is perfectly optimized to bind that phosphate moiety. And this way, we can make a 10 billion member library where every member of this library has intrinsic binding affinity to basically bind the phosphate species. And so this effectively targets all of our antibodies to a particular site on our target of interest. And this should drastically improve our chances of success at isolating these reagents. And by analogy, could we also in the future develop similar synthetic scaffolds tailored to other types of post-translational modifications? And this would provide us with a very powerful and novel synthetic antibody toolkit to rapidly generate these reagents to study the importance of these modifications in biology and disease. And so when I first started this project, the grand challenge was how do we explicitly recognize a phospho residue? And just I've depicted here schematically the three most commonly phosphorylated residues in proteins: phosphorylated serine, phosphorylated threonine, as well as phosphorylated tyrosine. And when I started this work, there was no structure of a phospho-specific antibody to kind of guide this work. I should say now there are 5 of them, four of which are mine. And this structural knowledge is extremely important in advancing this technology. And so I took a step back and thought about it and realized that, well, a lot of proteins, such as enzymes, must recognize phosphate to perform their function. And one common motif that most of these proteins use to recognize this phosphate, or this general anion, is what's known as a nest motif. And so what this is effectively a region of the protein, 3 consecutive amino acids, that the backbone main chain of the protein is oriented in such a way that we have 3 consecutive amide residues, all which are oriented in perfect orientation to make hydrogen bonds and recognize the anion of interest. And so I performed a computational search of all existing antibody structures to look for whether there's a similar motif in antibodies that we might be able to exploit to engineer a binding pocket specific for these types of anions, namely the phosphorylated amino acids. And I was quite surprised to identify quite a large fraction of antibodies which had a similar motif as this nest motif. So it's shown right here with these consecutive amino acids all oriented in such a way to contribute hydrogen bonds to, in this case, an aspartic acid, an anionic amino acid. And so to better characterize this mode of interaction and to see whether I could engineer this binding pocket to selectively recognize one of these sites of modification, I synthesized the genes and expressed this antibody in bacteria. And the first question I asked, well, does this antibody bind to phosphorylated species? Does it have some kind of weak intrinsic binding for that? And so I performed what are known as enzyme-linked immunoadsorbent assays, or ELISAs. And briefly what this is we can take our target, bind our antibody to that target, and then using a secondary reagent, detect the amount of antibody which are bound to our target. And so this allows us to determine whether any given antibody binds our target. We can introduce a different variation on this, which is known as a competition ELISA, where we can add various soluble competitors to compete with the antibody for binding its various target. And so this allows us to compete off the antibody against any given target to look at the specificity of a given antibody. And so what does this data look like? Well, briefly on the next couple slides, you're going to see on the y-axis a binding signal. And on the x-axis, gradually increasing amounts of the soluble competitor. So in the absence of no competitor, one would expect no change in binding signal. However, as we increase the concentration, we should see a decrease in binding signal. And by curve fitting, we can estimate the binding affinity for the various species. And so the first question I asked was, well, how well does this antibody bind to its original aspartic acid-containing peptide? And you can see here it binds quite well with a KD of around 20 nanomolar. And then I looked to see, well, if we change this aspartic acid to the unphosphorylated forms of serine, threonine, or tyrosine, what happens? And we effectively observed no competition, and so we've completely knocked out binding affinity. However, I was really excited to see that when I looked for binding to the 2 phosphorylated species, namely phosphoserine and phosphothreonine, I observed very weak binding on the order of around 5 micromolar, suggesting this binding pocket is indeed functional. It's just likely not optimized for the phosphorylated residues we were interested in. But this did give us a great molecular handle to further design in specificity for. And so looking at the original structure of the antibody, one can identify a number of antibody side chains which likely confer specificity to what type of anion this binding pocket is recognizing. And so I went through and made a series of antibody phage display libraries with desired chemical diversity in this region and performed the selections I talked about earlier to identify antibodies which bound selectively to phosphoserine, phosphothreonine, and phosphotyrosine, and then characterize the resulting antibodies. And so just as a frame of reference, again, this is our parent antibody scaffold that we started with, which binds quite weakly to both phosphoserine and phosphothreonine. After extensive characterization and isolation, I was able to isolate a phosphoserine-specific scaffold that does not cross-react with the unphosphorylated form of the protein, as well as a scaffold which effectively is indistinguishable at recognizing the phosphorylated form of serine or threonine and effectively does not bind to any of the unphosphorylated amino acids, as well as a phosphotyrosine-specific scaffold, suggesting tremendous modularity in this initial antibody and this motif in general. And so to better delve into and understand the structural basis of this recognition, I solved the X-ray crystal structures of all of these antibodies in complex with their phosphopeptide target. We wanted to take a picture and see what was actually happening in the antibody that allowed it to selectively recognize the phosphorylated amino acids. And so this involved a series of steps: expression in a protease deficient bacterial strain, a 3-stage purification, crystallization, as well as ultimately solving the structures. And this analysis was quite surprising and quite satisfying in that it revealed that these phosphopeptides, shown here as a magenta kind of lined across the antibody, which is shown underneath, bind in a linear bifunctional manner. So there's one region we could classify as our phosphospecific binding pocket, which effectively serves as that PTM anchor region that I was talking about earlier. And the second part can be thought of as a general reader domain, which effectively reads out the sequence context for this modification. So this allows us to tell where in the protein this modification is occurring. And so if we zoom in on this particular PTM anchor region to identify chemically what's happening and how we're able to obtain this highly efficient recognition, we can see in the case of the phosphoserine-specific scaffold a number of hydrogen bonds and a very dense hydrogen bonding network which mediate recognition of the phosphoserine, as well as a histidine perfectly positioned to make hydrogen bonds with the phosphate group. In the case of the phosphoserine threonine-specific scaffold, again, a different structural motif, but again, a highly dense network of hydrogen bonds, as well as a beautiful electrostatic interaction with this arginine right here. And in the case of phosphotyrosine, we observed a third motif to recognize this phosphorylated amino acid. So this is all great. We can begin to understand molecularly how we're able to recognize this side chain, but what can we do with this structural information moving forward? What does this enable us to do? Well, again, this bifunctional mode of recognition allows us to make a general analogy, and let's say we'd like to make antibodies against multiple different sites of phosphorylation. So we could effectively fix this PTM anchor region in our antibody scaffold. And so this way, every antibody we're making has an intrinsic binding pocket for the phosphorylated side chain. And then simply diversify this reader domain to allow us to rapidly make antibodies against different sites of phosphorylation in proteins. And so I generated 2 antibody phage display libraries with over 10 billion members based on both the phosphoserine as well as phosphoserine threonine scaffolds. And then piped those in vitro phage display technology to allow us to rapidly generate phosphospecific antibodies against various immobilized targets. And so really what this enables us to do is basically generate a knowledge-based platform that allows us to continually learn and improve this process and continually generate better phosphospecific antibodies, and in the future, other types of post-translational modifications. So we've designed and tested and validated this antibody scaffold, and then I generated a number of antibody libraries which now allow us to rapidly generate phosphospecific antibodies with high affinity and specificity. And we continually learn from this process by performing biophysical characterization of the antibodies, which allow us to make improvements in this initial designed scaffold. I can also sequence the antibodies we get out, as well as solve structures to improve the chemical diversity of this initial antibody library, as well as improve the recovery of these important reagents by carefully optimizing the selection conditions, such as concentration and times, to rapidly enable the generation of multiple phosphospecific antibodies. So as an initial proof of principle, I chose 10 sites of phosphorylation in a series of different proteins and generated biotinylated synthetic phosphopeptides against each of these targets. And then performed a series of selections with my phage display libraries and isolated 68 antibodies against these 10 targets. And then performed a series of ELISAs to validate, well, are these antibodies indeed selective for the phosphorylated form of the peptide, or do they recognize the peptide in some other context? And so I performed a series of ELISAs, and so on the y-axis here is the binding signal, and on the x-axis are different antibody clones. And so you can see in the case of, say, this antibody, this one raised against peptide 1, there's indistinguishable binding to the phosphorylated peptide as well as the unphosphorylated. So this would be something we don't want. But namely, importantly, in 7 of the 10 cases, there was at least one antibody which is highly selective for the phosphopeptide, and that only bound the peptide in its phosphorylated state, suggesting these scaffolds are indeed working to direct our antibodies towards the phosphorylated epitope. And when I looked against all 68 of these antibodies, namely 51 of the 68 antibodies were exquisitely phosphospecific. And this success rate of 75% completely blows away success rates purported by immunization and animal-based methods, which is on the order of 0.01 to, say, 1% at best. And more importantly, I've done this all in a single span of under a month to enable us to rapidly isolate these antibodies as well as begin to characterize them for defined functions. And so I wanted to take a couple steps further and determine whether these antibodies were specific for this particular target. So in the case of antibodies raised against peptide number 2, did they bind peptide 2, or did they just generally bind any protein or any species which had a phosphorylated amino acid on it? And so performed a cross-panel kind of large-scale analysis where I assayed the binding of these antibodies to all 10 of the phosphorylated peptides. And so I made all 51 of these antibodies, and here I'm showing representative data for antibodies which were selected against this peptide number 10. And you can see they bound only to peptide number 10, and they did not kind of cross-react with any of the other phosphorylated peptides, suggesting they are specific for this particular phosphorylated peptide. And so kind of a better way to represent this data is in kind of in a heat map format where we can normalize no binding to basically a black signal, high binding to a yellow signal. And you can see for these 4 antibodies, they're exquisitely selective for peptide number 10. And so when I looked against all 51 of these antibodies, on the top here, you're going to see all 51 of the antibodies which were selected against the peptide indicated above. And on the y-axis here, relative binding signal for all 10 of the phosphopeptides. Almost all of them were exquisitely selective for the peptide for which they were selected against, highlighting the fact that our scaffolds are generating not only phosphospecific reagents, but that can detect that phosphorylation in specific sequence contexts. And to take this one step further, I asked the question as well, these recognize the phosphorylated peptide, that's all well and good. Do they actually recognize the proteins? Do they recognize the protein when it is modified by a phosphate group? And performed a series of Western blot analysis to look at whether these antibodies also bind the phosphorylated protein. And here are results from 4 of the antibodies I made. And just briefly, looking at this particular antibody, you can see a strong binding signal to the original phosphorylated protein. However, when we treat that protein with an enzyme that rips off and enzymatically removes the phosphate group, we lose binding signal. And so in all 4 of these cases, there's an antibody that's exquisitely selected for only the phosphorylated form of the protein. And so really what this shows and validates is that these engineered scaffolds I've been able to develop have really enabled a robust platform for high throughput generation of new site-specific phosphospecific antibodies, and that we can automate this potentially using bacteria and rapidly generate panels of phosphospecific antibodies to begin to study phosphorylation on a much more global level than the traditional one site at a time method. And so in summary, I've introduced these synthetic antibody scaffolds that offer tremendous potential to rapidly generate functional antibodies with excellent biophysical properties. And in some work I didn't touch on today, in a collaborative effort with another postdoc in our lab, we were able to generate antibodies that can functionally modulate the activity of an enzyme. So these antibodies were actually able to activate a particular enzyme. To give you one example of potential future applications. Furthermore, I've generally introduced how structural understanding of antibody recognition really enables the rational generation of antibodies against entire classes of really highly important targets. And more specifically, I engineered novel antibody scaffolds which were specifically tailored to enable binders against phosphoserine, phosphothreonine, and phosphotyrosine, 3 really important and common sites of post-translational modification, and incorporated and applied these scaffolds to ultimately develop an in vitro platform that enables the rapid, renewable generation of phospho-specific antibodies. And in the future, this high-throughput phospho-specific antibody platform will really enable a comprehensive dissection of cellular responses that occur during healthy and disease states. And that will be able to take defined snapshots of multiple different phosphorylation events at any given time point. And ultimately, moving forward, I envision continuing some of this work to fully fill this synthetic antibody toolkit to enable us to have a very nice platform to rapidly generate reagents capable of detecting important sites of post-translational modification. And so in the context of protein phosphorylation, developing these improved methods to not only detect but quantify these phosphorylations will enable us to better identify what sites of phosphorylation are indeed functional, as well as beginning to describe and elucidate the role and potential therapeutic applications of this emerging class of phosphorylation events that actually occur outside cells on the surface of these cells. And extending much of this technology to begin to study other types of chemical modification, what information is kind of encoded by these modifications, particularly several post-translational modifications that are intimately tied to disease and metabolic diseases such as diabetes that have been shown to alter protein function and cellular responses. And inherent to a lot of these, there are no antibodies which allow us to detect and quantify these sites of modification. And so we effectively have no way to understand what these are actually doing to the cell and, more importantly, to the proteins. And so there are a number of questions generally to be asked, namely how these post-translational modifications also alter protein function, as well as this work will ultimately reveal new avenues for therapeutic intervention. And finally, more generally, I envision extending much of this kind of synthetic antibody and protein-based scaffolds to ultimately develop rational methods to engineer protein-protein interactions to now not learn something about the cell, but control what the cell is actually doing. And so this can take a number of different forms, developing these modulators to regulate the activity of these PTM regulating enzymes, as well as potential cellular receptors, both detecting and modulating the activities of oncogenes and things that go haywire in cancers, as well as ultimately potentially designing and building synthetic circuits which can sense and respond to different cues. And ultimately, this work will position my lab at the forefront of several areas that of protein engineering and molecular recognition, as well as systems biology and drug delivery to ultimately make fundamental discoveries in both cell biology with a strong translational application. And just as closing and acknowledgments, I'd like to thank my postdoc mentor, Jim Wells, as well as another postdoc in my lab, Nathan Thompson, who's been really essential in teaching me X-ray crystallography and helping me learn this new skill set, as well as various other members of the Wells lab, both past and present. My collaborator, Dev Sidhu at the University of Toronto, who I went to kind of learn initial phage display and antibody engineering methods, as well as my collaborator, Bill Delgrado at UCSF, and a former graduate student in his lab who assisted with some of the computational work. And last but not least, my funding source is the Life Sciences Research Foundation for a postdoctoral fellowship and the NIH NCI for a R01 on a different project that I haven't had time to talk about today. And last but not least, all of you. Oh, yeah. Yes. So that's actually a really cool story. So the question is, what about the fifth structure of the phospho-specific antibody that I mentioned earlier, and whether that, which came from a chicken, exhibits a similar mode of recognition of the phosphorylated side chain. And it actually does. So it came from a group in the UK that was immunizing chickens with these phosphopeptides and trying to identify an antibody from chickens that bound to a particular site of phosphorylation. And they solved the structure, and the chicken immune system actually uses a very similar mode of recognition in the same place as I've been able to design. So it does suggest we might have stumbled across a natural mechanism that antibodies have been using, just no one knew about. Yeah, exactly. So it's, let's see here. Yeah. So the nice part is, so this is all the site that I've engineered to be specific is kind of off to the side here. So it's contained within a single CDR loop, kind of highlighted here in red. And then I'm only diversifying specific regions of the antibody that I know can tolerate significant sequence diversity. So I'm not completely randomizing every position in every CDR. I've kind of focused the diversity. So all of the initial targets were 17 mers that basically have the phospho residue in the center. And so this effectively, I figured, enabled me to get 2 shots on goal. So we could bind the sequence upstream or the sequence downstream of the phosphorylated sequence.
You used a naturally occurring phosphate binding motif to attach a PTM outcome. I just wondered if you could speak to, in the context of other potential post-translation modifications, do you have your eyes on other motifs there that you could use as anchors in those cases? Are some going to be more complicated than others? I imagine glycosylation is kind of its own whole ball game. So I just wondered if you had sort of some view of that.
So I envision kind of taking a two-pronged strategy. So there are a number of natural proteins that have evolved to contain pockets for various post-translational modifications. So there's a class of proteins which bind acetylated lysine, for example. And they all have a very similar motif in that having basically a couple hydrophobic residues positioned in such a way to enable selective recognition of that acetylated. So there are some natural occurring motifs that I could use as a guide to do that. Some other stuff moving forward would be to just kind of discover new motifs as well, to kind of, in that we can, by understanding enough about antibodies and where they can tolerate sequence diversity, take strategies to design completely new motifs that might be selective for these sites of post-translational modification. And I should say, in the case of glycosylation, the glycan I'm interested in is about the size of tryptophan. So it's just a single glycan. It's not these extensive, complicated structures that people are used to.
I really good talk. Since you've been selecting this from antibody, I'd like to get your opinion. How do you, differentiate compared to an aptamer-based select method, and why you choose this, and can you make a comment with respect to the aptamer-based?
Oh, so aptamers. OK. So I guess aptamers are emerging. So the question is, what about aptamer technology? So aptamers are these RNA or DNA molecules, depending on what you choose, and basically trying to make secondary and tertiary structured DNA and RNA molecules that can do the same thing antibodies have evolved to. So the idea is to somehow select these DNA or RNA molecules that are selective for particular targets. I would say aptamers have been shown to work pretty well for small molecules and obtain pretty exquisite binders for specific kind of small chemical molecules. I think the jury is still out in terms of as a general recognition tool for proteins and kind of protein-based detection reagents.
So you did really fantastic job for selectivity. So what's your future goal for improving sensitivity?
Yeah, so I guess I should say, so the affinities of these initial antibodies are not great. They are selective in certain contexts. The affinities are on the order of a couple hundred nanomolar. And probably part of the reason are inherent initial limitations in the library design, in that I took a kind of quick and dirty strategy to introduce chemical diversity in this reader domain. And so there are lots of non-functional amino acids, such as cysteine or stop codons, which basically reduced the potential of our libraries to identify high affinity binders. And so by better learning from both the structures as well as the sequences I'm getting out, I can then inform this next generation library, which will hopefully routinely generate higher affinity binders.
Are there any other questions? Okay, one more there.
Right. So the question is whether they recognize the folded form of the protein versus the denatured form kind of that is seen in Westerns. I've tested one or two by immunohistochemistry, and they are fairly not as specific as I would like, and I think the problem is initial affinities that I'm getting. And so since we know the sequences, I can go back in and kind of further improve the affinities of these antibodies, whereas if it was an animal, I'd kind of be stuck with what the animal gave me. So.
Well, thank you again, JT, for a very nice